Greetings to everyone. Thank you for joining me today. My name is Chien-Ming Lo, and I'm honored to share with you our research on the relationship between microstructural variation in inhibitory interneuron across the human cortex and the topographic organization of brain activity as recorded from magnetoencephalography, or MEG. Here is a concise overview of the key aspects covered in my speech. I will quickly delve into the rationale behind our research, emphasizing the significance of structure-to-function relationship and highlighting the critical role of inhibitory interneuron. Next, I will explain the methods employed in our study, as well as the results we found. Finally, I will conclude by summarizing our findings. To better understand how the brain functions and processes information, it is essential to unravel the intricate relationship between its microstructure and the overall function. Previous study has described a robust functional hierarchy that situates different cortical regions along a unimodal to transmodal axis. Studies also try to associate the functional hierarchy with underlying microstructure that is revealed by T1-T2 ratio map, potentially indexing myelin content, which can also be associated to the principal component of brain-specific genes expression covariate matrix across the cortical areas, implying the linkage between the molecular variation, structure, and the functional organization. Here, we try to be more specific about the molecular variation. In particular, we have focused our attention on inhibitory interneurons, a specific group of neurons that play a crucial role in regulating and modulating neural activity within the brain. Unlike their excitatory counterparts, inhibitory interneurons exhibit significant morphological variation as well as various connectivity and intrinsic properties, providing a rich diversity for shaping the complexity of the brain. Our research aims to establish a potential microstructure to function relationship by investigating the interplay between the variation of inhibitory interneurons and the cortical functional organization as revealed by several metrics of MEG source signals. We employ the combination of gene expression data from Allen Human Brain Atlas, which provides around 20,000 gene expression profile across the human brain, and the resting state MEG recording from Human Connectome Project, which captures the dynamic cortical activity at the macroscopic level. We select 100 interneuron marker genes from previous literature and leverage the gradient approach to analyze the transcriptomic data we select, allowing us to gain some insights into these molecular variations across the cortex. For the MEG data, we conduct source reconstruction by LCMV method and compute large-scale features that capture different levels of brain activity, such as neuronal avalanche and the Xen-positive complexity. This combination approach enables us to explore the intricate relationship between molecular profiles and the macroscopic activity pattern across the human brain. Here is the basic gradient construction workflow. We begin by calculating a gene expression similarity for each brain region. This matrix captures the similarity in gene expression patterns across the brain. Then we apply a normalized cosine similarity kernel to the sparse data to ensure robustness and accuracy. Next, we employ a nonlinear dimensionality reduction algorithm called diffusion map. This algorithm allows us to decompose several low-dimensional manifolds, often referred to as gradients. Each of these gradients represents distinct aspects of the gene similarity matrix. We will focus on the first two gradients as they explain the most variance in our data. As you can see, the gradient 1 follows an anterior to posterior axis that explains the most variance of the input matrix. And gradient 2 demonstrates a clear unimodel to transmodel division that has already been observed in the classic functional gradient. We perform categorical regressions to predict the gradient values using the 7 functional network as predictors. Not surprisingly, our findings reveal that the gradient 2 show twice better explanatory power compared to the gradient 1, according to the R-squared. Moreover, when we organize the second gradient values based on the functional networks, a clear pattern emerged. We observe a clear division from unimodel networks, such as somatomotor and visual networks, towards other more transmodel networks. To further prove the concept, we acquired the classic functional gradient from the HCP dataset and the histological gradient derived from the microstructural profile of the big brain histological dataset. We compare these cortical maps using the spin permutation test. The outcomes of this analysis reveal significant evidence of association between these gradients, suggesting the presence of a shared organizational properties. But we observe no such associations when comparing these gradients with the first interneuron gradient. However, our investigation did uncover some connections between the gradient 1 and the large-scale features of the MEG recordings. Here, we investigate 3 key features of resting state MEG. First, the neuronal avalanche, which serves as a measure of local excitability based on the occurrence of very high amplitude burst events. We observe a pattern where neuronal avalanche decrease gradually from the posterior to the anterior part of the brain. For this index, we found a negative association with the gradient 1. Second, Zempo-Z complexity, which quantifies the compression level of broadband MEG time series in each brain region. Our findings reveal that frontal region exhibit higher complexity scores, suggesting a greater level of unpredictability, where the visual area exhibit the lowest complexity level. This complexity pattern aligns with the gradient pattern 1. Lastly, the intrinsic timescale, which is derived from the autocorrelation window that reaches the cross point of 0. The pattern observed in the intrinsic timescale aligns with the complexity finding, but with sensory areas demonstrating the shortest time window. This cortical map also shows a positive correlation with the gradient 1, suggesting a link between the compressibility, autocorrelation, and the molecular variation captured by our interneuron gradient 1. In summary, our analysis revealed 2 primary underlying manifolds derived from the regional interneuron similarity matrix, each capturing unique facets of cortical organization. The gradient 1 exhibit a robust association with the resting state MEG features that provides insights into local excitability, complexity, and autocorrelation patterns. Gradient 2 demonstrates a more pronounced association with the organization of functional networks and also existing functional and histological gradient. These findings provide a bridge between neuroimage features and the underlying microstructural characteristics, specifically the interneuron expression similarity. Thank you for your attention. I'm here to acknowledge my supervisor, Dr. Neil Duncan, and our institute, GIMBIC, that makes this work possible. Thank you.